So if you are familiar with bioinformatics and Python, you should know about the Biopython library. So Biopython is a library in Python which is used for bioinformatics data analysis. So we are going to use the Biopython as well in our course for the data manipulation and data learning. So Biopython is powerful and easy to use bioinformatics library that allows developer to perform complex tasks related to your sequence analysis, database searching, and much more. So, first thing that the bioinformatics library, which is Biopython, can do is sequence manipulation. So, Biopython offers you robust and efficient method for handling your biological sequence, such as DNA, RNA, and protein sequencing. It allows easy parsing, indexing, manipulation of sequences, facilitating tasks such as subsetting, translation, and reverse complementation. Also, the file phrasing and formats. BioPython supports phrasing and writing various bioinformatics file formats such as FASTA, GenBank, PDB, BLAST, PDX, VCF, and much more. This capability simplifies the handling and analysis of biological data retrieved from different databases and tools. So, sequence alignment and analysis. BioPython often provides tools for the pairwise and multiple sequence alignment, which we are going to perform in our sequence analysis section. We can also use BioPython for phylogenetic, structure analysis, blast integration, bioinformatics algorithm development, and data retrieval. Features of BioPython. So BioPython often features sequence objects which will handle your DNA, RNA, and protein sequences, your analysis and visualization tasks such as pairwise alignment, protein structure analysis, and phylogenetic trees. Database searching. BioPython can be used to search various databases that are available on the internet, and also its integration with the another tools such as PDB, skitlearn, and other libraries as well. So sequence object. So a DNA sequence object, RNA sequence object, and protein sequence object is basically that is used in your BioPython libraries. Working with sequence and alignments, you can read and write sequences by using any file format like FASTA, GenBank, and can also be written using different libraries. Provides tools for pairwise sequence alignment like Needleman and Vinci algorithm. Biopython often provide a variety of method for multiple alignments such as cluster W algorithm, and also Biopython has many methods for performing common tasks such as reverse complementing sequencing, translating DNA to protein, and much more. Database searching. So BioPython Entrez module introduce you to search online databases such as PubMed, nucleotide protein, and numbers of others. So NCBI's basic local alignment tool, which is BLAST, is also used for comparing sequencing to find the homology between them. BioPython makes it easy to automate BLAST searches. Swissprot is a high quality sequence database. BioPython provides a module for searching Swissprot and downloading the sequences. So available tools and modules that are used in BioPython. The common examples that we can use are SecIO, Philo, Proteomics, and others. So conclusion, BioPython is a powerful and flexible bioinformatics library that makes it easy to perform DNA, RNA, and protein sequence analysis. Developers have give access to a wide range of biological databases and functional analysis using available in the library. With the new feature and update being added, the future perspective of this library are bright and exciting for bioinformatics community. So that's for the literature. That's for the talk that we can do. And if we move on to our VS Code section, here we can see we got first of all what we can work with. We can import our sequence into our BioPython library. So first of all, what we can do from bio is our BioPython module. So how can we install Biopython? So it is simple. We just need to install Python in your system and then write pip install Biopython. Install Biopython, and Biopython will be installed in your system. From there, you can import your bio library, and after that import the seq.io. Seq.io is a module that work with the sequences. Sequence input or output. Biosec is documented at biosec at you can find various range of guidelines from here and also from the book. So first of all, what we want to do is we want to open a sequence file which is named by sequence.fasta. So let me just go to the folder where I have put all these files, and here I can search for sequence.fasta. So here is the sequence that is provided here. So I am going to just load that, and I'm gonna name it file sequence.fasta. For record in, I'm gonna praise a file name which is in the FASTA format, and then just print the sequence. So here is the sequence. You can see it's a protein sequence. So how can we make a reverse complement of a sequence? That we can reverse complement our sequence. For example, if we have the adenine data, adenine base, we can change it to thiamine, and if we have thiamine, then we can change it to adenine. So from bio.sec module, we are going to import our sequence object and sequence class. Then we can create a DNA sequence. So I have just input ATGCG, and also input anything you want, and then just create a function of reverse complement. And after that, we are going to reverse complement that sequence, and then just click on the run button, and after that we will get our reverse complements. Here you can see here is our reverse complement of the sequence. So transcription, changing the DNA to RNA. So just like the transcription reverse complement, you will create your DNA sequence object. After that, you are going to give RNA sequence, which is going to DNA sequence, and then transcribe it to RNA. So here you can see adenine is adenine and thiamine is changing to uracil. So translation is changing your DNA and RNA to your protein. So here you can see. So you can see here we have just created our DNA sequence, and if we run this, it is going to change our DNA and our RNA sequence to our protein sequence. So sequence alignment. So this is just a basic overall BioPython functionalities of BioPython work. So by importing the align class and align module, you can create an object align dot pairwise aligner. After that, you can just click and input your first sequence and then second sequence. After that, you are going to align your sequence 1 and sequence 2, and then you are going to print your alignment, and here you can see you will get the result like this. You can also compare sequences in BioPython, from your BioPython library. You just have to import your pairwise module, and after that you have to import your sequence module because we are working in a sequence, and then we will get our sequence 1 and then sequence 2, and after that we can perform our pairwise global alignment, and then we are going to print our global alignment, and here you can see we have compared those two sequences and the score is available here. So that is pretty much it for the introduction of BioPython. You don't have to worry writing all this code for your practice. You can just download this notebook from my GitHub library, which I will give you in the description or somewhere else. So thank you so much for your attention, and see you on in the sequence analysis video.